Chapter 4, Biology of the Cell, Part 5. So, we're moving on to cell division. Cell division occurs when one cell divides to produce two or more new cells. And cell division is very critically important. It is necessary for development of organisms and tissues. It's involved in tissue growth. It's needed in tissue repair, in replacing old cells that are getting worn out. So it's critical both in, say, a child getting bigger and an adult just maintaining their body. Two forms of cell division. There's mitosis. Mitosis is a division in which one cell with 46 chromosomes produces two new cells with 46 chromosomes. The new cells are pretty much identical to the original cell in most cases. So these are somatic cells, various body cells that are being produced. Cells of the body, somatic cells, so most of them. Meiosis, on the other hand, is a process where you produce sex cells, sperm or oocytes. This time you start with one cell with 46 chromosomes, and it becomes four cells with 23 chromosomes each, and these four cells are genetically distinct from the original parent cell. Here is a karyotype, which is a way to image to view the chromosomes of someone. So if you look at this karyotype, see we have 23 pairs, 46 chromosomes, and this person happens to be male because they have one Y and one X. Cell cycle. Cell cycle is basically the various processes the cell undergoes during its life that often includes cell division. So we have the interphase state, which is what the cell goes through most of the time as it's living its life, and then the mitosis phase, the mitotic phase, when it's undergoing mitosis. So interphase is actually the phase that the cell spends most of its time. This is when the cell maintenance is occurring. This is when the cell's doing the metabolic activities it's supposed to do, doing the job it's supposed to do. Also during interphase is when the cell undergoes processes to prepare to divide. So there are three subphases to interphase. We have G1 phase, S phase, and G2 phase. G1 phase is the initial growth phase. After the new cell is produced from mitosis, it's a little smaller than normal. So that cell needs to grow, needs to produce new organelles to get to the size it needs to be to undergo its normal activities. And also during this phase, you have structures needed for DNA replication also being formed in anticipation for future phases. Now, it turns out many cells leave the cell cycle at this point and never divide because they don't divide. So neurons, for instance, muscle cells, another example, these are cells that don't normally divide, so they would leave the cell cycle at this point. S phase. S phase is when DNA replication occurs. This leads to the production of duplicated chromosomes. So it's chromosomes that now have two copies of DNA, and the two copies of the DNA are in structures referred to as chromatids. So the duplicated chromosomes have sister chromatids, two chromatids that are attached by a structure called a centromere. So for instance, here's an electron scanning view of chromosomes. As you can see, you got two sides of the X. Here's where the centromere is. This is one sister chromatid. Here's the other sister chromatid. Chromatids, again, have identical copies of the DNA. DNA replication, pretty straightforward. The DNA will unwind, opening up the double helix, and then DNA polymerase comes in and forms a complementary strand of DNA using one of the old DNA strands as a template. So the DNA polymerase comes in and does complementary base pairing to attach new DNA nucleotides together to form the growing new side of the DNA molecule. So you have two chains formed at the end of the replication, each with one old strand of DNA and one new strand of DNA. And you, of course, need DNA, deoxyribonucleotides, and the DNA polymerase. So again, DNA polymerase attaches to the DNA, boom, 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 brings in the DNA nucleotides, complementary base pairing, and you get a growing strand of DNA that's going to give you a lovely new double helix. In the off strand, going the opposite direction, this has lagging strands and little short pieces that have to get ligated together, and we're not going to go into that complicated process. All right, G2 phase. G2 phase is the second growth phase, and the goal here is to produce more organelles, more than the cell needs, to get the cell bigger than it needs to be, because what it really has to do is get big enough that after it divides, the two new cells are large enough to survive. And this is when the final preparations for the beginning of mitosis occurs with, say, enzymes that are needed to be synthesized. All right, then we enter. So we entered, so we are at interphase G2. Interphase G2 has two centrioles because, of course, these are important for this process. The nucleus with the chromatin, the relaxed DNA inside of it, and ready to go. Then we enter the mitotic phase, the M phase, mitotic phase, where we undergo mitosis, which is the separation of the nucleus. Actually, mostly talking about the separation of the DNA, and then cytokinesis, which is the division of the cytoplasm, the actual physical dividing of the cell into two cells. And it turns out cytokinesis and mitosis overlap during the mitotic phase. Mitosis has four subphases: prophase, metaphase, anaphase, and telophase. So, in prophase, beginning of mitosis, the nuclear envelope starts to break down. The chromatin condenses into the chromosomes. So we now have chromosomes instead of chromatin, so these denser, more solid structures holding the sister chromatids with the two copies of DNA. Nucleolus breaks down, nuclear envelope breaks down. The centrioles start to travel to opposite sides of the cell, and the centrioles start to produce spindle fibers that will stretch out from the centrioles toward the center of the cell where the chromosomes are. Metaphase. During metaphase, the chromosomes line up in a row along the equatorial plane. So you see it pretty much lining up in a row in the equatorial plane. The spindle fibers stretching from the centrioles will reach the chromosomes and attach to the centromeres. So the spindle fibers attach to the centromeres of the chromosomes during metaphase. Then in anaphase, the spindle fibers will shorten, and as they shorten, they will separate the chromosomes into their two sister chromatids, pulling the chromatids, now called chromosomes, toward opposite ends of the cell, toward the centrioles that are producing and now shortening those spindle fibers. So now each side of the cell is going to get a copy of each chromosome. Telophase. Telophase, the new chromosomes now arrive at the opposite ends of the cell. The nuclear envelope begins to reform around the chromosomes. The chromosomes begin to relax, become chromatin again. The nucleolus reforms. Mitotic spindle breaks up, basically returning to the way the cell would be during interphase. And of course, cytokinesis is occurring. Cytokinesis, division of the cytoplasm. So often during anaphase, you get the beginning of the cleavage furrow forming for cytokinesis, and then telophase, it's pretty obvious that the cell is breaking up as the cleavage furrows meet in the middle to fuse and form the plasma membrane of each cell. And basically this occurs through a function of microfilaments that are basically squeezing the plasma membranes together, forming a contractile ring. That's what is making that cleavage furrow. Cancer. Cancer is uncontrolled cell division. Cancer is when mitosis is no longer controlled in a healthy manner and instead produces massive amounts of cells. Cancer is caused by two genetic mutations to the DNA. Carcinogens, mutagens, whatever, are damaging the DNA of the cell, causing the cell to mutate. And usually there ends up being a series of mutations necessary to get cancer to develop. The first phase is what's formed is what's called a benign tumor. Moles are benign tumors. Benign tumors are relatively harmless because they do not invade and damage and destroy surrounding tissue. However, they can get quite large. So if this benign tumor happens to be in an inconvenient location, it could be dangerous. In this case, this 60-pound tumor was removed from a morbidly obese individual. It was actually in their abdominal cavity. But you can imagine if that was in the lung or thoracic cavity, that could lead to a lot of problems. All right, after the benign tumor forms, it will eventually, potentially, develop additional mutations to the DNA, turning it into a malignant tumor. Malignant tumors will now invade surrounding tissues, destroying those tissues. So this is now inherently dangerous, at least in that location where the tumor has developed. And then after the addition of even more mutations to the DNA, the malignant tumor develops the ability to undergo metastasis. So the malignant tumor is now able to send out individual cells that will spread to other parts of the body, developing tumors there and damaging the tissues in those other parts of the body. So again, lots and lots of accumulation of mutations to the DNA lead to the development of cancers. All right, it turns out we do occasionally have cells die. Cells can die through necrosis. Necrosis is basically an accident. Something harmful has killed the cells. You accidentally stab yourself with a knife, you spill acid on your skin. It's unexpected, undesired damage and cell death. Apoptosis, on the other hand, is programmed cell death. This is cell death that is desired and it is planned on. It involves destroying and removing cellular components in a nice controlled manner so it does not have too much of an adverse effect on the cell. And it is initiated by ligand receptor signaling. So certain chemicals, ligands, bind to receptors on cells, causing the cell to begin the process of apoptosis. This occurs during proper development when we were all fetuses. At one point, we had tissue between the fingers between our fingers, giving us little webbed hands. And then over time, as the fetus developed, those tissue between the fingers underwent apoptosis and disappeared. Also can be used, apoptosis as a way to get rid of harmful cells, can be a way for the immune cells to cause our own body cells that are infected by a virus to destroy themselves in order to stop making that virus. Can be useful to cause apoptosis in cancer cells because cancer cells look a little different from our normal cells, and sometimes immune cells can detect that. So again, apoptosis, programmed cell death, the desired, planned for death of our cells. And that is it for this lecture and this chapter.